thank you so much for inviting me to give this distinguished lecture, and I also like to thank you for the kind introduction. And actually, I do have many fond memories about Brookhaven. I used to come here in the 90s for data collection at the NSLS at the time, and really we spent a lot of days and nights collecting data here. So it's but I haven't really been back since today, and really been quite impressed with the NSLS2 and all the new exciting things that are happening there. So it's really great to see that. And so we certainly would also be quite keen using the facility at some point. So so as was mentioned here, I sort of started out in X-ray crystallography and using really computational methods to assist with determining structures, particularly in the area of refinement. And then in the mid-90s, decided to really switch my focus on working on a particular biological problem or area. And this one area which I decided to work on concerns, generally speaking, vesicle fusion, and more specifically, the release of neurotransmitter. And so what I'd like to tell you today is tell you about some of our fairly recent results. Some of them are using X-ray crystallography, and other results are using electron microscopy and also some optical microscopy. So we're really using now a variety of different technologies and methods to understand this particular problem. And so I'd like to maybe begin in context of this lecture, giving you a sort of brief introduction into modern neuroscience, and of course can't be very comprehensive. So what I decided to do, give you sort of a brief visual history of what you might call molecular neuroscience. And maybe we can have the lights down a little bit. I think it's a little bit, if that's possible. And so there were, these are probably some of the earliest drawings of the human brain on the 16th century. And at that point, the brain was really not well understood in terms of what it would actually consist of. The concept of neurons was not known at the time, and primarily what was noticed were the sort of very high degree of vascular structure in the brain. So it was very a lot of blood vessels. But what actually was there in this brain matter wasn't clear, because it wasn't really visible to the techniques at the time. And so it was really only after it had become possible to essentially stain a subset of neurons and to image them under a microscope, optical microscope, that the concept of neurons was created. And that was really the work by Cajal. And this is one of those rather beautiful drawings that he created, essentially looking at brain tissue under an optical microscope. But the important point here was that he used a labeling technique that was invented by Golgi that only labels a subset of the neurons. So suddenly a problem that was intractable, because the brain matter is so dense, it's impossible to really see anything under a microscope. Now, with this labeling techniques, a subset of the neuron became visible, and he was able to create drawings which quite realistically show what we now know about the neural networks. Now, of course, this is at a relatively low density level given the staining method, but what you notice already, there's some neurons, or some parts of neurons have these tree-like structures that interdict trees, and others these are axons that have that don't have these features, and then every once in a while the two cross, and And then when you look closely, sometimes he was actually drawing a little dot here. So it was a thicker structure that emerged where the axons and the dendrites were contacting each other. And of course, today we know that's characteristic of a synapse. It has it's a very sort of dense feature characterizing a connection between two neurons. And so, and it was then about 50 years later that it was possible to actually image some of those synapses by electron microscopy. This is just one example here. Now, it's relatively difficult here to see anything unless you look very closely, but what people recognized are small spherical objects that are called synaptic vesicles. So that's the first sort of visualization of what we now know of synaptic vesicles. That took some more developments in electron microscopy and also how specimens were prepared to actually image those synapses in more detail. And this is just one example here. It's basically, it's a electron micrograph from a high-pressure frozen synaptosome. And as it becomes clear from that image, is that there are, again, these synaptic vesicles, and some of which are closer to this, what is here, a membrane. And so this is the presynaptic neuron here, part of the axon, and this is a postsynaptic neuron, part of a postsynaptic neuron. In this case, rather characteristic density that contains receptors and other important molecules. And so now, moving forward now in technology, now this is using cryo-electron tomography right now, again, of synapses. In this case, this is work from the Baumeister group. And we can see those vesicles even more clearly right now, and also with the tomography was able to reconstruct in three-dimensional image, and this is one example here showing essentially three-dimensional surface rendering of synaptic vesicles as they're docked to this, we call the plasma membrane here. So, and just to give you a little bit of an idea of a scale here, so these synaptic vesicles are about 40 nanometers in diameter on average. So, so this is where we are today in terms of cryo-electron tomography of intact synapses. And now another important development happened also in the 50s, and that's work by Bernard Katz, who was mentioned here already. So in his pioneering work, what he did was to look at the electrical properties of neurons, more specifically at motor nerve endings. And he was stimulating them electrically and then looking at the reaction from those neurons. And he observed that there was a response at certain times. And when he plotted the distribution of these amplitudes, they were centered around a certain unit. So that led Katz to propose that the release of neurotransmitter occurs in discrete jumps, or as people call it, that they're quantized. Now, quantum here is not a single particle, unlike in physics. It's really a bunch of neurotransmitter molecules, but the point is that the amount of transmitter is a certain discrete entity, or the average amount. And of course, what he then realized is that this corresponds to the fusion of individual synaptic vesicles with the plasma membrane. Now, this was visualized some years later by Heuser and Rees, who again used electron microscopy, but this time they imaged the synapses and synaptic vesicles here before stimulating the neuron, and just a few milliseconds after stimulation. And look what happens here. Many of these have fused with the membrane. You still see kind of the shape, part shape of these synaptic vesicles as they have merged or fused with the membrane. Okay, so this is a clearly very sort of instructive demonstration of the idea that when synaptic vesicles fuse with the membrane, they release neurotransmitters. Now, of course, these electron micrographs, they don't really tell us much about what is really doing all this work, because it's not just membranes that are there, but there's many, many proteins involved in the process. So just to give you some idea about the complexity, so this is these are models based on mass spec proteomic analysis of synaptic vesicles and of entire synaptosomes, so which essentially contain the entire presynaptic part of the synapse. And so already when you look at a single synaptic vesicle, there are literally 100 or so different proteins, different types of proteins in those vesicles. And when one actually looks at the density of these proteins, it's roughly equivalent to the density of an enveloped virus. It's a very high density, relatively speaking. But unlike viruses, where typically these proteins are arranged in some symmetrical fashion, as far as we know today, there's no such symmetry in synaptic vesicle, but rather they are distributed, although it's not clear if there may be some sort of order to them, and that remains to be discovered. But the point is there's many different proteins, and we only know the function of a subset of these proteins. And there's the majority, we don't really know what they're doing. And likewise, in the cytoplasm of presynaptic neuron, there's many proteins that we don't know what they're actually there for. We don't know, again, the role of some of them. And also what we don't know is exactly what complexes they form and how they're interacting and how they're changing dynamically as the neuron is undergoing stimulation. So this is sort of where we are in a way at the moment. It's sort of at a molecular level, we're kind of at the level that before Cajal actually invented a method to really image individual synaptic connections. It's a very complex, very dense set of proteins, and so just looking at electron tomograms doesn't really tell us much in detail, with some exceptions clearly, but generally about all these different proteins. Now, what I'd like to do here today is to talk about some of our work, which specifically involves a type of release that's response of stimulation of the neuron. And so this is also called evoked synaptic vesicle fusion. And briefly, the principle now is that when a neuron is stimulated, there's a change in the voltage potential created across the presynaptic membrane. And this change in voltage potential will in turn open transiently calcium-dependent, voltage-dependent calcium channels. And it turns out that the calcium concentration in the synaptic cleft is much higher than in the cytosol. So what happens is when these channels open, that there's a rush of calcium into the presynaptic neuron, and it's that calcium that acts as a signal to actually then cause fusion of synaptic vesicles with the target membrane. So that's the principle of evoked release of neurotransmitter. Once they're fusing, the neurotransmitter is diffusing into the synaptic cleft, and they ultimately will bind to postsynaptic receptors and then trigger a stimulation of the postsynaptic neuron. So this is what's called a chemical synapse. It's couples the electrical change of the membrane into a chemical signal, which then in turn produces another voltage change on the postsynaptic neuron. So now, so the general principles are known, of course, for many years, but there's sort of many mechanistic questions which are still very much unclear. For example, what is the mechanism of calcium control, and how is it possible that this can all happen within less than a millisecond? So for biological membrane fusion, this is very fast. There's other membrane fusion events which, for example, viral fusion entry that happen on a much lower time scale. So this is the sort of fastest known membrane fusion event. And in addition to that, it is highly regulated. So the calcium will go from a very low probability of fusion at resting calcium potential to a higher probability of fusion upon stimulation of the neuron. So how is it made possible that the system essentially acts like a switch? Sort of, in a way, you can look at it like a transistor. Those of you who are electrical, you know about transistors. So they're basically, in a way, voltage-controlled transistors. They produce a very large signal upon change of the potential across the membrane. So how is it possible that this is so precisely controlled? So now, as I already mentioned, in terms of the complexity of the system, many different factors involved, at the moment we have to reduce the system to really begin to understand how it works. And so we and others have been using a combination of bottom-up and top-down approaches to uncover the molecular mechanisms. So what do I mean by that? So by now it's known based on genetic and physiological and biochemical experiments what some of the key players are. So we can begin to understand this sort of minimal set of proteins and how they actually function. And then we can go back into the neuron and ask the question, what else is there that might be important? And by essentially probing what we learned from those studies, the reduced system, and test the models in the neuron, and then make the models more complete. And ultimately, of course, we would like to know how it exactly works in a neuron. But as I said already, the resolution of electron-tomography is not sufficient to really tell us exactly where all the individual proteins are. So let me begin by introducing to some of the key players in this process. And I promise to make it sort of as simple as possible, but I have to introduce you to some of the key players here. So first of all, at the center of this machinery are the so-called SNARE proteins. What are SNARE proteins? They're a group of proteins, and I will show you some structures later, that tend to form complexes with each other. And so in the case of synaptic neurotransmitter release, there's typically one type of SNARE in the synaptic vesicle and another set of SNAREs in the plasma membrane. So when the synaptic vesicle docks to the plasma membrane, then this forms a complex. Now, because it crosses or juxtaposes membranes, we call this a trans-SNARE complex. So it's between membranes. Now, there's an important step until it reaches this point here, and it's generally called docking and priming. We'll come back to that later, what this means, or at least the understanding we have of this step. And now this complex here alone does not explain why there is calcium dependence of this process. These proteins on their own are not calcium dependent. So what is required now is a calcium sensor that comes in, then combination with calcium will then trigger fusion. So somehow the calcium sensor and this complex act together to make it a regulated process that's triggered by calcium. So at that point, the complex is now in the same membrane. One calls this a cis-SNARE complex. Now, the story isn't over, because what now happens is that these complexes get disassembled by a machinery called NSF. It's an ATPase that takes this complex apart into its individual components, and it makes them available for another round of formation of synaptic vesicles through the process called endocytosis, and I won't say much about it here. And then they can begin again forming trans-SNARE complexes and so on. So the point here is there's a cycle of assembly, fusion, and disassembly of this SNARE complex. So these are three steps, and I will tell you today in more detail now about formation of this complex, calcium triggering, and the disassembly itself. So let me begin with the SNARE complex itself. So this sort of summarizes what we know about it today. We now know that the complex is formed, and when it forms, it pulls the membranes together, sets the stage for fusion, as is illustrated here. And many years ago, we determined actually the structure of part of the SNARE complex and showed it's a parallel full-helix bundle. So that supported the notion that it forms this parallel arrangement, and since there are transmembrane anchors in both membranes, that means it has to pull membranes together. Now, so intuitively, that suggested also in combination with other data that it is the complex formation that actually drives fusion. Does it make sense physically? Well, there's now experiments that illustrate that energetically it could make sense, because the energy to actually that is released by this formation of this complex is about in the same order of magnitude as to initiate the step for fusion. And this is based here on one hand by taking the assembled SNARE complex and pulling it apart with optical tweezers or magnetic tweezers, work from Zhang and Yun. And so they showed that taking this complex apart in this, these tweezer experiments requires roughly this order of free energy. And now in contrast to actually, or for comparison now, the energy that's required to initiate the fusion of membranes, more specifically the stalk formation, is estimated roughly to be about 40 to 90 kBt, and this is based on experiments where layers of membranes were desolvated, and then the extra reflectivity was measured, and it was observed at some point there is formation of so-called stalks, and that allowed them to give an estimate for the energy it's required to actually form these stalks, and the idea behind it is actually that membranes have to be desolvated in the first place to allow membrane fusion to occur. So the point here is that the energy that is released by one SNARE complex in assembly is in the same order of magnitude to trigger fusion. So why energetically it makes sense to propose this model. Now, of course, there's generally more than one complex required because this is still a little bit short on actually the energy for this process. In addition, there's additional energy required to actually trigger a complete fusion event. And so typically one would estimate maybe between 2 and 4 or 5 complexes required, or even a few more. Now, let me move on now to the calcium sensor. So this process of formation of SNARE complex is calcium independent. So as I already mentioned, it requires a calcium sensor. And one such sensor was discovered by Tom Südhoff. It is a, these are proteins that have single-spanning membrane domains, and typically 2 calcium binding domains are called C2 domains, structure of which one is shown here. And what was already early on recognized that they can do 2 things. They can interact with the membrane, more specifically with anionic membranes, in a calcium-dependent fashion, and they can also at the same time interact with SNARE complexes. Now, the importance of these molecules was really demonstrated in experiments from the Südhoff lab. For example, this one here, that this shows a wild-type response of a wild-type neuron upon stimulation. It's essentially a postsynaptic current that is measured here. And whereas this happens when the synaptic tag, when one is genetically deleted or knocked out, that produces, that essentially eliminates this fast response here. And then moreover, what was shown later is that there's a correlation between calcium affinity and calcium sensitivity. So there's a correlation between the molecular signal or the molecular underpinnings of this process and the actual sensitivity of neurotransmitter release. Now there's one more rather biological side, and apologize for all the biology here for those of you who are not familiar with this. But again, that's sort of, I think I have to introduce you to some of those in more detail. So this is another factor here that is called complexin. And I'm just showing this here because it illustrates that the genetics here, in this case, complexin deletion, is not as severe as the deletion of synaptic, which essentially completely disrupted the process. But there's still a significant effect on deletion of complexin. And so it's clear that also is important in the process, okay? And so the question in the field has been, for many years right now, how these factors, synaptic fragments, the calcium sensor and complexin and the SNARES, how they all cooperate to trigger synaptic vesicle fusion. Now, when I ask this question, of course, the first question is this really a sort of sufficient system that has calcium sensitivity? So I'd like to tell you first is a reconstitution experiment where we took those individual components and tried to reconstitute calcium-triggered fusion. I will then tell you about structures that are relating to this, these interactions, and then speculate about mechanisms of the process. So reconstitution. So for us and others, it's been really rather essential to perform biochemical and biophysical studies with reconstituted proteins and to try to understand these relatively simple systems and then move on to more complex questions. And so this shows the assay we developed, and there's other variations of different types of assays, but at a fundamental level, many of these assays consist of creating proteoliposomes. So these are lipid vesicles with reconstituted proteins. And more specifically, we use two classes of vesicles. One that's tethered to a surface, and using a method that TJ Ha developed some years ago. And so these vesicles, in our case, contain those SNARES which are in the plasma membrane. And so they're meant to mimic the plasma membrane, although very approximately though. And then the other class of vesicles are with reconstituted synaptic proteins, so it's SNARES involved in or included in synaptic vesicles as well as the calcium center synaptic fragment. And then we can add additional factors such as complexin to the system. We now let the two classes interact. So these are tethered. We add the others. They form complexes. And then we inject calcium into the system and we watch fusion. Now, how do we watch fusion? We use we use dyes, fluorescent dyes that are encapsulated in these vesicles, and they're initially in a self-quenched state. And when fusion happens, volume increases, and that increases the fluorescence signal increase. So that's how we can detect fusion events in these assays. Let me show you an example here. So this is this shows the fluorescence intensity from a vesicle that's at a docked typical site. And this vesicle doesn't fuse either without or with calcium. This is an example where a vesicle has docked, and then in this calcium-independent period at 0 calcium, fusion happens. This happens every once in a while. There's a calcium-independent fusion event. This is an example where fusion happens after the injection of calcium. So this is calcium-triggered fusion. How do we know there's calcium there? We use a dye that comes with the calcium injection, so we know we can detect the arrival of calcium. So what the advantage of this assay is it discriminates between simple vesicle association or docking and fusion events, or what we call content mixing. These are just some examples here. This is fusion independent of calcium, so at 0 calcium. There's occasional events, but not very many. When we inject calcium to the system, there's a pronounced increase in the probability of fusion. And there's some very fast events and relatively slow events coming later. So that's very encouraging because it demonstrates that those two factors alone, so the synaptotagment and the SNARES, are sufficient to have calcium dependence. But as you can see, it's not very efficient. It's only about 1% of those that are fusing right away when we inject calcium. So that clearly is not very physiological. Now, what has now become really important for us is that as we now made the system more complete by adding additional factors into the reconstitution, this efficiency has dramatically increased. So first, when we introduced complex into the system, just mentioned to you, it has a large increase in the amplitude, so it's roughly a factor 2 or 3 higher than without it. And that really also tracks the results in neurons, as I showed you earlier, that when complex is genetically deleted, that reduces the amplitude about half. So this is sort of actually quite good agreement with the physiological result. But it's still, in our minimal system, it's still not very efficient. It's only 2% now. But things have improved more recently now. So again, this is the same graph here with complexins, so it's about 2%. When we add additional factors, and I'll come back to what these are in a few minutes, we're now up to 10% fusing right away. So this is becoming more respectable now. And also, we actually don't need a relatively high calcium concentration anymore. This happens also with relatively physiological calcium concentrations now in the tens of micromolar range. Now, what do all these other factors do here? So this shows the reconstitution is becoming more complicated, but very briefly here, what we do is there's a step where we convert the initial complex into a complex between syntaxin and MUNC18, and then we add MUNC13 and these synaptic vesicles, and we then observe fusion. Now, I will come back to NSF later. This is the ATPase, which I mentioned in the introduction, that disassembles the SNARE complex. So what it actually does in this case, it disassembles this initial precursor complex and allows this other complex to form with the MUNC18. Okay. But then we wash it away, and we now then initiate the reaction by adding the synaptic vesicles and MUNC13. So I'd like to say a few words about what these two factors you do. And this is now concerning the assembly process itself. So far, I sort of implied that when we simply take those SNARE complexes, they form, this is much better, by the way, thank you. They form a complex, and it sort of implied that it would always get it right. Now, when I moved to Stanford, a few years after I moved to Stanford, in collaboration with Stephen Chu, and I'm sure you know Stephen, especially from his time as the energy secretary. And that was before he had moved to Lawrence Berkeley Labs at the time to become director of Lawrence Berkeley Labs. We started a collaboration, and one of the questions was to actually understand vesicle fusion using single molecule methods that Steve and others had developed. And so what we did was to put labels in different parts of the SNARE complex and ask the question, do we actually get SNARE complexes correctly assembled? And so the idea is to put labels based on the crystal structure that should produce high FRET if the complex were properly assembled. But if it was improperly assembled, for example, an antiparallel configuration, we would get a low FRET signal. And of course, initially we were expecting to see mostly high FRET. But much to our surprise, we saw actually almost 50-50 a properly parallel SNARE complex. On the other hand, we saw intermediate or low FRET populations where the accepted dye fluorescence is much lower than what expected if it were properly parallel assembled. Now, at the time, we had a hard time publishing this, and eventually we published it in PNAS, and also sometimes called previously not published in Science, but anyway, we finally published it. But what was really not clear at the time is if this is true, if in fact when snare complexes form, that it's not always the correct assembly. And so many years later, then, and this is work by Brandon Choi in my group now, he discovered that when we use those additional factors, like the Monk 13 and the Monk 18, we now always get parallel snare complex. So this is just one experiment here using labels on 2 different snare components of the complex, and we primarily get high fret. Now he went on to put labels on different parts of the snare complex, and again he got high fret. So the point is that these factors, what they do is they promote the proper assembly of the snare complex. So what they do is really they, in a way, act as chaperones to promote the proper assembly, and this is summarized here. And so at least in part, that's their role. There may be other roles as well, but certainly based on our sort of reconstitution experiment at the moment, at least, that's their primary function in this sort of minimal system. So that's was a long way of saying that we can basically assume once these factors are there, we always get parallel snare complex. But what we don't know yet, or we didn't know, was really how the snare complex now interacting with the calcium sensor and complexin and so on. So this is now work by Zhong Zhang or John Zhu, was published last year, of the complex. It's really a super complex now between the snare complex at the center, synaptotagmin, and part of complexin. So this is, shows the 2 C2 domains of one synaptotagmin molecule here interacting with the snare complex. And we'd actually determined that part previously in the previous crystal structure already, so we knew that part. What's new here was really that we were able to introduce complexin into this, and what we saw to our surprise was another interface between the second synaptotagmin molecule and both the snare complex and complexin. So we really have 2 ways that these synaptotagmins can interact with the snare complex, one that doesn't involve complexin and the other one that does involve complexin. So, and this is interesting because, as I mentioned to you already, when we take complexin out of our reconstitution fusion experiment, it still kind of works. We can now explain this because this interface is independent from complexin, whereas this one, of course, depends on complexin. So we basically take half of the machinery out when we remove complexin, but it still kind of works. Now, this interface here between synaptotagmin and snare complex without the complexin is actually calcium independent. That was also another big surprise. So now we'll come back to that in a few minutes. Moreover, this complex, for those of you crystallographers who, of course, one might be worried about effective crystal packing contacts, we do have very different crystal forms of this and see the same interface, and it's identical in all these different crystal forms. And we also have other evidence, and we did a lot of functional experiments, both with our fusion assay as well as with neuronal cultures, and I won't go into the details, it's all published here. And essentially all these experiments sort of really confirmed the notion that this interaction is important at some point during the process of release. In particular, we suggest it's very important in the pre-fusion state, but it also might be important at a later stage. And we'll come back to that later. What about the other interface? So this is the one that involves the snare complex as well as this synaptotagmin molecule, part of it, and this part of complexin here. So what's interesting when we solve the structure is one of those discoveries. When solving a structure, one immediately recognizes that it makes structural sense because look at this helix here of complexin, it's part of complexin. It's continued into a helix in synaptotagmin, and it's perfectly well aligned, so it's essentially a continuation of this helix. And of course, this interaction is dependent on complexin. When complexin is taken out, this interaction doesn't take place. So again, we did functional tests, and again, we found that disrupting residues or taking away complexin disrupts this interface and reduces or eliminates release of neurotransmitter. So that's where we are. So we know that this sort of minimal set of proteins, the SNARES, synaptotagmin, and complexin, are a calcium-dependent system, as we showed in our assay. We have a structure of the core part of this complex now, but we really like to figure out what is the molecular mechanism itself upon calcium binding, upon calcium triggering. So now before we can speculate about that, we have to actually figure out how is this complex located between the membranes. So again, this is the structure here, but this doesn't tell us how it's sitting between the membranes. Or maybe does even the conformational change once it's in the membrane. It's quite possible that, of course, this is in the soluble complex. When it's in the membranes, it could be different. And then also there's questions how many of these complexes there are, a lot of controversies in the field, and if they're interacting. So all these are open questions. So I'd like to just show two possible models here, and these are really meant to be a sort of more thought experiments and don't take them too literally here. So this is, be taking two complexes side by side with the transmembrane domains at the center here. And the structured parts, so the helices and the beta sheets, that's our crystal structure, and the unstructured parts that we really don't know exactly what their conformation is. So these are clearly models here. And we also don't know exactly what the membrane separation is, but we'll come back to it in a few minutes. Now, important part of this model is that the synaptotagmins are close to the membrane. This is, for example, is a region that's known to interact with anionic lipids. It's a very basic region. So this is, in fact, in our model, it's close to the membrane, close to the plasma membrane here. And the same is true for the other synaptotagmins, also reasonably close to the membrane, although I've drawn it further away here. The calcium binding loops, which are the ones in yellow here, which in this form, again, this is in the absence of calcium, so they're actually sort of pointing peripherally rather than into the membrane. And that's important. We'll come back to that later. So they're not right in the membrane. But this is only one possibility out of many possibilities. Again, there could be more complexes here rather than two. It could be 3 or 4 or 5 or 10 arranged around this center here, right? So, and that is a possibility. So this, we're not suggesting it's just 2 here. It's just for simplicity, I only drawn 2. But the other theoretical possibility is then rather having 2 synaptotagmins interacting with one complex. It could actually be one synaptotagmin interacting with two SNARE complexes. So this is the other arrangement, which I call the bridge arrangement. So now we have a one synaptotagmin C2b domain bridging 2 complexes via the 2 interfaces. This one here, the other one is here. And based on the structure we have, that requires that the transmembrane domains are on opposite sides of this supercomplex. Now, at first, this might be counterintuitive because it would seem to exactly do the opposite to what we expect fusion should do, where transmembrane anchors would squeeze the membranes together and trigger fusion. So this is on the opposite side. So that's, at first, it's a little counterintuitive. But now, remember that this complex, in the absence of calcium, we don't get much fusion at all. And so somehow this complex is inhibiting fusion. And if it, for example, rearranged in this fashion, it would actually be quite intuitive why it would do that, because it would, again, the transmembrane domains would be on opposite sides, so they would possibly be able to fuse membranes because the protein complex is in the center between the membranes. It would essentially block fusion. Does it make sense? Now, we have one important piece of evidence that suggests that this, in fact, might be what's going on. And this is now using electron cryotomography. So what we did was to take the proteoliposomes we used in our assay, in our fusion assay, and imaged those proteoliposomes and their contacts, okay? And these are just some first data. This is a collaboration with Wolfgang Baumeister's group in Munich. And so again, these are the same vesicles we used as in our fusion assay. We know they're functionally active. And so we put them and deposited them on an EM grid and then carried out EM tomography experiments. This shows this one projection image here. The larger vesicles correspond to the plasma membrane mimicking vesicles. The smaller one correspond to the synaptic vesicle mimicking vesicles. And every once in a while, what we were able to observe are connections between these membranes. This becomes a little clearer here. This rendering here, surface rendering, the blue is the membrane density, and then the remaining difference density is in red. So these are clearly suggest that these are proteins sitting between membranes. And of course, given that we know what we put into the system, the only factor that can be in those contacts are those proteins which are reconstituted, so synaptic tagmin complex and SNARE complex. Now, what's interesting with these experiments, we observe a number of different morphologies. There's sort of loose contacts and tight contacts and long contacts. But the separation between membranes is somewhere between 70 angstrom and maybe sort of 30 angstroms. So it's significantly far apart. And looking at one of them more detailed, this is actually the actual micrograph here, or the particular orientation that shows the contact very clearly, again illustrating this separation between membranes. And that actually, when the membranes are that far apart, fusion cannot happen. Because for fusion to happen, they have to get closer than 10 angstroms. And so they're pretty far apart. But they're close enough to envision that once something happens with whatever is sitting here, then fusion could happen at a very rapid time scale. Because everything is in the right place to then trigger fusion. It's also clear that what we're looking at here is not a lipid stalk because it's too far apart. So therefore, it has to be a proteinaceous complex. And in fact, when one looks at the volume, approximate volume, it could be 2 or more complexes fitting into this. This is where we are very preliminary, so we're now trying to get a lot more tomograms and ultimately do subtomogram averaging and really trying to figure out exactly what's in this density here. Now, so this comes into the model itself. So let's a moment step back and let's look at one complex and make it a little simpler. Sitting between the membranes, and I just illustrated that this complex keeps the membranes apart and effectively inhibiting fusion. And in fact, there's supporting evidence for this notion that this complex inhibits fusion because there's a certain class of mutations of synaptic tagmin that when they're introduced into the neuron, they have dominant negative effects. In other words, even though there's wild-type endogenous synaptic tagmins in the neuron, the introduction of these mutations will essentially abolish release. And we interpret that by saying that if there's complexes such as this with a mutation that is unable to bind calcium, then having just a few of those present would not allow membranes to fuse because they're keeping the membranes apart. And furthermore, we showed that the tripartite interface is essentially for these dominant negative effects, so they're supporting this model. So again, so the idea is that we have a inhibited state of the system, and we very much believe that the crystal structure we determined reflects this state. And then what happens when calcium binds? And so the idea is that calcium binding will do something dramatic to the synaptic tag. Either it might completely dissociate from this complex by interacting with the membrane, as illustrated here, or it might go to a different part of the complex and then initiate fusion. And so these are just some of the two possibilities here. So it's either dissociating from the complex, from the SNARE complex, and going to the membrane, or going to a different part of the SNARE complex, or both. So that's the first step. And then what has to happen then is actually fusion itself. And so again, this is the released state now. So now the membranes can get closer to each other. And also what will probably happen at that point is full zippering of the SNARE complex and full fusion. Now, so there's a number of possibilities here how these different steps might promote fusion, and probably it's a combination of all of them. And so full zippering of the SNARE complex, that could be one major component, trigger fusion. It would also be synaptotagmin-induced membrane bending. And we actually believe it's further away from the fusion site, essentially providing leverage for that process. And then finally could also be some sort of membrane bridging. So all of them probably will act together to then trigger fusion at a very fast time scale. But of course, these are all models that ultimately we hope to visualize by capturing intermediate states in a fusion reaction. Okay, so coming back now to this overview. So I talked about the assembly, and I talked about triggering. Let me say a few words about the disassembly itself, and I'll make this relatively brief because I'm running a little bit late here. So I mentioned already that after fusion has happened, then these SNARE complexes are recycled in the neuron, and this happens many, many hundred times or more. And it's actually, so this ATPase is one of the highest energy consumer in the brain, it turns out. And it does, what we know right now is essentially keep going around and disassemble SNARE complexes. Once they're disassembled, they basically are providing the energy for subsequent fusion events. So in a way, it's sort of like an energy storage system that provides new photogenic snares for other subsequent rounds of fusion. So NSF was discovered by Jim Rothman many years ago. It was shown to be important as a factor involved in vesicular trafficking in cells, and later was realized or recognized to be also important for release of neurotransmitter. Now, it's an ATPase, so it has ATP binding domains, and this particular ATPase has two different domains called D1 and D2. D2, we determined the structure. We independently built Weiss, again, many years ago, and then also the N-terminal domain, we determined independently structures of it. So again, this is sort of ancient history by now, and then there was many, many years where we in other labs tried to get a structure of the full-length NSF, as well as, of course, structure of the complex between NSF and SNARES and so on. And it was really only recently we were able to achieve that. Before I do that, let me say a few more words about the actual process itself. And so what NSF requires actually is another factor called SNAP. So this is essentially an adapter protein that forms an intermediate complex with a SNARE complex, which in turn then interacts with NSF. And so this super complex here, SNARES, SNAPs, and NSF is called the 20S complex. And upon ATP hydrolysis, this complex, or this machine, disassembles the SNARE complex. And actually, biochemically, you can beautifully show that. This is one particular assay that we used, and there's other assays also. But it demonstrates that it's such a beautiful biochemical system, but yet it has been so difficult to get a structure until recently. It was really only with the advances in electron microscopy, cryo electron microscopy, we were able to determine the structure of this complex at sufficiently high resolution to actually be able to map all the components in this machine. So this is the structure we published 2 years ago. It's roughly at 7.6 angstrom resolution. And we do know crystal structures on the individual parts of this complex. So this actually illustrates how important crystallography still is, because we actually, in this case, we could not get, certainly at that point, an atomic resolution structure based on EM alone. We're now doing a little better. We're getting to 4.5 angstrom also, but still, knowing crystal structures is really still essential for these much more complex systems that show significant heterogeneity and therefore certain lack of resolution. So this is now showing the EM map. It's a sharpened map. Clearly you can see how beautiful we can fit individual components into this map. And, but it's relatively low resolution. Now, what are some of the insights you obtained from this structure? The first big surprise was at the top here, where there's a SNARE complex in the center, and then these SNAP adapter proteins wrapped around the complex. And this is something we would have probably never predicted, that these adapter proteins are wrapping around SNARE complex in the opposite hand. So the SNARE complex is a left-handed superhelical bundle, but the SNAP's going around in a right-handed fashion. So it's the opposite sense. And it sort of might suggest that as this process now proceeds, and again, this is the starting point of the system. It's in a non-hydrolyzable condition, so it doesn't dissociate the complex. That as hydrolysis proceeds, maybe one of the effects might be that this tilt might increase and therefore start to unwind the complex, literally. So that's the idea here. And again, we're hoping to capture these intermediates in the future. The other interesting finding was about the NSF-ATPase domains, which are down here. And we found that they have a significant asymmetry, especially the D1 ring that goes from a split washer conformation to an open flat washer condition starting from ATP into ADP. And these sort of asymmetrical transitions have now been observed in quite a few others of these AAA ATPases. So it seems to be common evolutionary phenomenon that upon hydrolysis, there's significant asymmetrical changes in these rings or domains, and that in turn would then trigger the particular process, in this case, disassembly of the SNARE complex. So I'd like to leave you with this picture here of first ideas about the mechanism. So there's the initial formation of that adapter complex between SNAPs and SNARES. By the way, they're actually in the membrane, so that's, although the structure itself is without the membrane parts. And then NSF comes along, forms this 20S complex, and then what probably might happen is this unwinding combined with some sort of threading into the pore here that will ultimately lead then to disassembly of the complex. Again, this is a model that we're hoping to capture these images or these intermediates in future studies, and probably primarily using electron microscopy in this case. So let me at the end just sort of summarize in a graphical fashion where we are right now. So we're, of course, talking a lot about the SNARE complex, and talked about how these other factors, the MUNC18 and MUNC13, that they are involved in the proper regulated assembly of the SNARE complex, which in turn then sets the stage for formation of the supercomplex with the synaptotagma. So this is the pre-fusion state before calcium arrives, before calcium binds to it. And then I did talk about the opposite phenomenon, the disassembly through this ATPase that is then dissociating the individual parts, and the whole cycle can begin again. So as you can see, we've sort of populated this cycle, but of course there's much more to be done, and really need to figure out exactly what happens upon calcium triggering and how the disassembly works of this machinery, and that will have ultimately also have more broader implications for this general family of fusion proteins as well as for these particular types of ATPases. So I'd like to just in the end acknowledge some of the co-workers and collaborators which are key in these projects. So John determined these structures of the synaptotagmin complexes, SNARE complexes, and the physiological studies were done in collaboration with Tom Südhoff. And then the electron microscopy experiments was performed by Quetic Gripson in my lab in collaboration with Yoshiyuki Fukuda and Wolfgang Baumeister's group. And the reconstitution, most recent contributions were from Ying Lai and Brandon Choi, who did these single molecule experiments. And then finally, the 20S complex, Minglei Zhao, who's now professor at the University of Chicago, assistant professor, and then more recently, Ian White is now involved in the project. We've improved the resolution most recently. And so the earlier work was done in collaboration with Yifan Cheng as UCSF. So I'd like to stop here and thank you for your attention. I'll be happy to answer questions.
Wow. Are there questions? There's someone who can bring up the house light there.
Yeah, there's one question over there. I can see that. Very good questions. Based on our reconstitution experiment, we know that it just takes a relatively brief period of time, maybe a minute or so, until they form complexes which are fusion competent. So just based purely empirically in these experiments, it's a relatively rapid process. And we do typically use a protein number density that's comparable to that has been observed in actual synaptic vesicles. And so clearly it's a process that does happen on a reasonably fast time scale, but it's possible that there's additional factors present in the neuron and synaptic vesicles that might further accelerate this process. I mean, there's the large majority of proteins, you have no idea what they're doing, and maybe they're there to sort of pre-organize the system, but we don't know that. I'm just suggesting that's a possibility. Yeah, no, it's so the first when the neurotoxins you're referring to are introduced, what they do is they politically cleave some of the SNARE proteins, so they are actually unable to form complexes in the first place, and that explains why neurotransmitter release is inhibited, because they cannot form complexes in the first place. Essentially, they're separated from the transmembrane parts. And so now, in terms of other factors that might cause disassembly, well, mutations in NSF, and there's relatively few known because they're so lethal. So there's a type of mutation that has been studied, especially in Drosophila, that essentially causes paralysis again, because once NSF is inhibited, once it's mutated to an inactive form, recycling cannot take place, so the system stops to work. So we know that NSF is essential for the process, so manipulation of that will disrupt release of neurotransmitter, and that's also how it was originally discovered, actually, because when it's modified, some of the initial pioneer experiments by Rothman, when it's modified, then it essentially disrupts the fusion between vesicles. So.
Are there any information on these proteins, or the 100 or so that you described earlier, from neurological diseases in humans, for instance, that might give you some clues as to functions?
Relatively few. There's, for example, one of them that I mentioned today, however, the MUNC18, certain mutations are known to be related to epilepsy. But beyond that, relatively few so far have been mapped to particular genetic disorders. And maybe one explanation is that mutations that actually cause a loss of function would be so lethal that they actually don't occur. But there's many factors sort of are related to the system or that are interfering with the system. For example, certain neurodegenerative factors are known to interfere with this machinery that prevent assembly, for example, of the SNARE complex. So a protein that's related to Parkinson's, alpha-synuclein, for example, it's been shown that in its sort of pathogenic form, it would disrupt, or it does disrupt the assembly of the SNARE complex. So that, again, so these are other factors that interfere with this machinery that obviously have dramatic effects. So.
What is the estimated time scale for this unwinding process at the lab?
It's so biochemically, when you look at the disassembly and the test tube, it's sort of in the order of seconds. So it's not a super, I mean, it's not milliseconds, but it's reasonably fast process. Right, so there's a couple of possibilities. I mean, the time scale of sort of seconds, you could envision capturing the intermediates by freezing at different stages after initiating the reaction, for example, by uncaging of ATP. The other idea might be to design mutations which would essentially trap the system into certain intermediate states. So, so these are some of the avenues which we're pursuing in this case. So.
SNARE complex alpha-helical bundling looks a little bit like influenza hemagglutinin alpha-helical stock in that trimer. Is there any similarity in the fusion?
Very good question. I think there's a lot of similarities. I mean, first of all, there is, of course, the sort of a large conformational change from opposite membranes with the sort of, in the case of HA, they're in opposite membranes, and then it undergoes a conformational change, and basically the whole thing just, that point forms a complete bundle. So it's very much similar, analogous to what the SNARE complex is doing, again, going from the sort of open, splayed open conformation into a fully formed helical bundle. And also, I think the other, what I talked about earlier, also the fact that these complexes are sitting between the membranes initially, and then upon some signal, pH in the case of HA, and here it's calcium, then something dramatic happens, a conformational change, dissociation of components and so on, and fusion happens. So I think there's definitely some similarities here. So the system is sort of in a precursor stage that allows us to undergo fusion. Now, there's also differences, of course, in that the viral fusion is much slower time scale, and also it's generally involves maybe different intermediate states in the process. So there's clearly also differences, and the system here has evolved to be very rapid and also extremely sensitive to calcium. So that's, I think, where some of the differences are. But fundamentally, the sort of use of energy from complex formation, I think that's the common similarity here.
Way back.
Is there any degradation of the actual factors in the times that they're used in test tube? Did you find that they became degraded over time?
Well, yes. I mean, I think, and that may be because we have some sort of sparse set of proteases in our test tube. So they're generally good for a couple of days, sitting at 4 degrees, so yes. But eventually things will degrade, so. But certainly it's relatively stable, certainly on a time scale that you can easily set up these experiments, both the fusion experiments as well as, of course, tomography experiments and so on.
Let's thank our speaker again.